Welcome to tonight's virtual clinic. The Macular Society have been running these webinar events now for over a year, really since the summer of last year, when we could no longer do our regular face-to-face seminars, roadshows and conferences. And so these have proved to be extremely successful. And as I've said probably many times before, we shall keep them going well after the pandemic is over. So it's great to have so many people here with us tonight, and thank you very much indeed for joining us. Now, as usual, we have an expert, a talk this evening from one of the most eminent ophthalmologists, scientists in ophthalmology anywhere in the world. There will be time for your questions at the end, so please do use the messaging box function to put your questions in, and I promise that I will do my very best to get to everybody's questions at the end of our guest's talk. So if you're a regular at these events, you'll have heard many talks now on the causes of age-related macular degeneration. In fact, of course, you'll know that the cause isn't known yet. We don't really know what causes macular degeneration. We do know that there are what we call risk factors, biological or lifestyle factors that are more common in people with macular degeneration. Certain genes, for example, whether somebody smokes, whether somebody has high blood pressure or a diet that's low in antioxidants, all of these are risk factors. But the biggest risk factor of all, and the clue is in the name, isn't it, in age-related macular degeneration, is age. The older we are, the more likely we are to develop AMD. So what is happening in the aging process that causes this? My guest tonight is working very hard to find out the answer to this. Professor Luminita Paraoan is a molecular biologist and one of the most eminent scientists in the world in this field. Indeed, she was recently named as one of the world's 100 most important women in ophthalmology. She has won many prizes for her work from revered institutions in Europe and the US, and she now leads the Ocular Molecular Biology Group at the University of Liverpool, indeed a group that she set up. She is Vice President of the International Eye Research Group, and we are very fortunate indeed that Luminita also has now become the chair of the Macular Society's own research committee. This is the group of scientists which who evaluates the applications to our research funding program and makes recommendations to our trustees as to which projects we ought to fund. Luminita's own team has received grants from the Society in the past for some of her work, and tonight she's going to tell us a little bit about it, particularly about her mission to understand how the aging process affects the eye. So, Professor Luminita Paraoan, a very warm welcome to the Macular Society Virtual Clinic this evening. Thank you very much indeed for joining us. And I'm going to hand over to you now for you to give us your talk. Luminita, welcome. Hello.
Hello. Hi. Thank you, Kathy. Can you hear me? Is everything all right?
Yes,
yes. Hi, and hello, everybody. It is really very strange to speak without seeing you all, but I am very happy that you were able to join us this evening, and I do thank Kathy for her very generous words, and obviously I'm very grateful for this opportunity, which is indeed lovely to be speaking to so many of you, and it's a particular fitting day to have this webinar today, as I'm sure you will know. It's a day in which we try to raise awareness of a condition which affects almost half of the people suffering from age-related macular degeneration. That's Charles Bonnet Awareness Day. Now, I was asked today, and I thought I would speak, for 20-25 minutes to you about two major questions. And if I could start sharing my screen, there will be only very few slides, and I'll be talking throughout them. Let's see if we can do this here. Just a second. So share screen. Okay, if you can, Emma, if you can help me, enable me to share the screen.
We think you should be able to, Luminita. We'll just.
It says no.
Yeah, she should.
Okay. You should now be able to do it.
Okay. Right. Okay. Yeah. Yeah. So let me know if you can see it. Yeah.
Yes, we can. That's perfect.
Right. So you see it. Right. So these are the two very big questions that I sort of try to say a few words about this evening. The first is, what happens to our eyes as we age, and why does macular degeneration occur? And these are huge questions, and some that clearly I cannot address them in any way near comprehensively tonight, but I'll emphasize one or two main points, take-home messages. Not only these questions are big, but the actual conditions that they address in relation to eye are very complex ones. And indeed, the figures that are sort of estimates of in terms of macular disease sufferers are quite sobering ones. In 2018, there were almost 1.5 million people in the UK alone affected by macular disease. And this is by far, as you know, the biggest cause of sight loss. Worldwide, it's estimated now to be in the range of 200 million people sufferers, and these include age-related macular degeneration, with a further about 30 million people affected by some sort of sight impairment. We're talking about 3 times the entire UK population worldwide being affected. In the UK, this is predicted to double as the current numbers are, and to reach by 2050 about 1.3 million people affected by macular disease. And this is equivalent to 400 new cases per day, per each day. It brings huge costs, both personally to the communities where we all live in, and to the UK, indeed up to 2 billion annually. It is estimated that it costs the UK economy and the health services to deal with this. It is an area where I could not just go further without saying that in terms of research that is aimed at solving and understanding and developing treatments for these diseases. So this research is structurally underfunded, and it is indeed both macular disease and AMD, the funding allocated to these are proportionately less than their prevalence. And it is here where Macular Society plays such an important role in this research environment in the UK, and not only. These are big problems that are not going away, and especially with an aging population, with increase of life expectancy, and with the increase in what we wish to have a healthy, longer lives, obviously are more and more of acute importance. The question that has arisen for quite some time, and it's still valid today, is why is it so difficult to find a cure? And this is, I'll show you, well, I'll tackle a few problems here to hopefully indicate where does this complexity comes from. For now, I would only mention the fact that if we are to think of the eye is an organ which is made up by numerous types of cells, and it's a really very complex organ. We're thinking, so it comes from the diversity and complexity of tissues that make up the eye, a well-functioning eye. It comes from the intricacy of processes that occur inside those cells and being mediated by those cells. It comes from the multitude of factors that influence our eyes and our eyesight. It also comes from the difficulty in studying eyes. It's not as easy as taking simply pieces of this particular organ, like could be, for example, taking a skin biopsy. And it comes from the complexity of developing efficient interventions. So now if we move into more scientific, if you want, aspects of the macular disease, and I'll focus on age-related macular degeneration. So obviously I set up in this question, and I will go by describing referring to the words in its very name in the reverse order. So we'll talk a bit about degeneration, about macular, and the aging influencing processes within the eye. So first, in terms of degeneration, this is describes a state or process of decline or deterioration. And this term is used generally to describe conditions, diseases that occur in the eye, not only, and very frequently in relation to state of deterioration of brain. And in those cases, we refer to it as neurodegeneration. Now, as I'll show in a couple of minutes, within the eye, we talk about indeed a neurodegeneration for when we look at or refer to processes that occur in cells of neuronal origin or very similar to those that are in our brains, and also to degeneration in general that affects cells other than those that are neurons or neuronal related cells. And I think at this point, probably the easiest will be if we'll look at a very well-known image, sort of presented, I'm sure, in many of these talks in different ways. Please don't worry about the actual writing or anything to, that is written here. I only want to use this as a preamble to remind ourselves that the eye has been often described or compared with a camera, or being referred to as a window to the brain. Again, probably taking a bit from the fact that it has a lot of cells that are similar to the ones that are found in our brain. And I also refer to it very often as 3 balls in one. And these 3 balls can be thought of an external one that is protective, shown here in blue, a middle one, which is barely seen here. And again, just don't need to actually look at details, just, follow, what I'm describing. And this middle ball, it's almost complete. It has a hole, if you want, where the pupil in front of the eye is, and we see part of it as the iris, the bit that gives the color of our eyes. And finally, an inner ball, which actually is even smaller than the others, so it's a lining, and it is in this lining where the retina lies. And retina is in itself a very specialized tissue formed by numerous cells. There are over 50 types of cells making up the retina, organized in a fantastically structured way. And I'm going to show you a section through a retina, simply to exemplify this fantastic structure. So see, this is just a cross section of a very small tissue here at the back of the eye, which just exemplifies these various layers, and it's a very, very neat and complex and indeed fascinating structure that makes the retina. The retina has lots of cells that are similar to the ones in the brain, and it also has some specialized cells that sense the light and they indeed have inside them components that react to light. And then somehow they manage, with lots of processes, intricate processes that occur inside those cells, to send signals, to transform, if you want, the light into signals that are sent to the brain through the optic nerve. And this is where the images and our vision actually is formed. I wanted to still look at this eye image here to make a point which actually occupied us more than an hour discussing, myself with some members of my group and with members of the Wirral Society of the Blind and partially sighted only last week, when we find as a fascinating discussion showing simply the fact that macular, it occupies a very, very small portion of this part of the back of the eye. And indeed, if the eye as an organ is quite small, the human eye has roughly 2.5 centimeters, an inch across, and it's not a perfect sphere, by the way, as I'm sure you know. The macula is around 3, maximum 5 millimeters across. So this whole lining that I've spoken about, the inner lining of the eye, it's formed of macula in the center and a lot of this peripheral retina. And the reason we spoke a lot last week about, and it's just a point that was very well made by all participants in that very informal meeting, is that how much more potential is there to be using this peripheral vision. However, the disease, age-related macular degeneration, occurs in this central area. And why is this area most affected? Because it is the exact part of the retina where the light is focused by the lens. And this is where indeed the highest acuity vision is supported by, and is supported by having a certain density of those cells shown here on the right. The very far back in our eyes lies this so-called photoreceptor cells, which are, as you know, rods and cones. And they are throughout the retina, but they have different types of densities in different areas, with the cones being mostly represented in this area. You will also remember that rods are the cells that ensure our vision in dark and in dim light conditions, where cones are the ones that help us see color and fine detail, but they are the ones that need a lot of light intensity. Now, it's probably sufficient to talk about neuroretina for the time being. We're moving on to a tissue which is, I think, even more impressive in terms of function, because indeed it is essential for maintenance of this very impressive structure itself. And it's nothing else but a single layer of cells at this level, which, again, for the more curious ones, I'll say it's called retinol pigment epithelium. So it's an epithelial type of cells, a bit similar to epithelia that is on our skin, but very different in many other respects. It is pigmented and it supports the function of the retina, hence its name. It is widely recognized now that although the vision, for the vision, the structure of the neuroretina is needed, the very first changes, and probably the most progressive, if you want, changes that occur in the eye with age, those that lead to age-related macular degeneration, occur first at the level of this tissue. And you could ask, as we all the people ask, why is that? This is quite widely accepted, this fact, although we now know that there are changes elsewhere, for example, in the tissue underneath, even further back of these epithelial cells. And I would want to make another point here about these retinal pigment epithelial cells. As they are depicted here in this drawing, this is one of the very first drawings of the retina, now simplified, that I've made more than 20 years ago. I've tried to highlight how big, bulky each of these cells are. And you will notice that on one side, they have these finger-like structures, which do nothing else but embrace the tips of those light-sensing cells, the photoreceptors, the rods and cones. And these cells are so big that only one of them embraces about 30 to 50 of these photoreceptor cells. In order for the retina to function, this embrace has to be very tight. And it has to be tight because it has a very specific function. And that is that it's performed throughout life and every day of our lives. And it starts in the morning when we first open our eyes, when these finger-like structures snatch the so-called spent parts of the photoreceptor cells, all the ones that contain all the proteins, all the components that have been used the previous day in ensuring our vision. They're snapped, they're broken down from the photoreceptor, and they need to be recycled and renewed in such a way that the actual light-sensing cells renew themselves. Without this function, just as a curiosity, we would not be able to see more than 7 days in our life. So this is how essential the function of the cells is. I've already alluded by giving this example to the fact that these cells have a fantastically intense metabolism, fantastically intense activity every day of our life. And this high metabolism in biology comes with not only with very high energy consumption, but also with producing a lot of waste material and also a lot of byproducts that, if they're not cleared properly, add to the damage of the cell. And this is indeed exactly what happens at the level of these cells. And it's because of these and other essential functions, as we won't have time now to mention, this retinal pigment epithelium was also called a nursemaid to the photoreceptor. And, it was estimated that throughout life, it's, in relation to the example I've just given, about 100 million rod discs, each of those RPE cells. So high metabolism, they're absolutely essential, very specialized. And on top of all this, these cells have a very, very limited ability, if at all present, to regenerate. So those cells that we are born with have to last us throughout our life. Now imagine that, or I mean, it's not really difficult to imagine, but think of the fact that pretty much everything with a lot of use and intense use obviously starts giving signs of wear and tear. And this is why at the level of this monolayer, we probably see one of the best examples of aging and aging functions in the body. So no wonder that, aging will have an increase in age, will have effect on how these cells function. And this is why, going all the way back now to describing age in terms of risk factors for developing age-related macular degeneration would be quite easy to understand why aging is actually the highest risk factor. And indeed, in roughly half of the cases of AMD sufferers, there isn't a genetic component. It's simply that the aging process affects these cells in such a way that it deteriorates their function. And with that, it deteriorates or declines the function of that very fantastic structure that lies above it and which absolutely depends on the well-functioning of these cells. I would refer to two more functions of these cells to understand then an example which I've chosen from our research to exemplify what these age-related changes and how they occur and what they are. The first one is that, as you'll see in this image, you'll notice very easily, these cells are what we call polarized. They're one side is different from the other. So they have very different faces. On one side, the one that faces the neuroretina are these finger-like structures. On the other one, it comes in contact with yet another very specialized structure. It's vasculature at the back of the eye. So every cell is in itself a factory that makes a lot of components. These are, mostly of them are proteins. These proteins are all made up in certain specialized compartments of the cell, but they have to be transported, trafficked, they have to be sent in specific locations inside the cell. In other words, if a certain protein type is required in a function in relation to these finger-like structures, it has to be sent there, specifically there, efficiently and timely there, as opposed to some other proteins that function on the other side of the cell, or indeed on the sides of it. So that's the mention of this trafficking, the sending of proteins and components where they are needed. The other point I want to make is that you will notice, again, it's just suggested in these drawings, the fact that there is some filling around these cells, the bottom of them, and indeed between them and the neuroretina. And this filling, it's also very important for how cells sense each other, how well they're attached to each other, and it's directly contributed by proteins, by components that are made inside the cell. So in other words, what I'm trying to say here is cells produce components that are needed for their inside processes, but also a lot of components that are needed for this filling outside. And this is true for other cells, for majority of cells in the body, that is. But again, in the case of the retina, we talk about very specialized components. Which then now brings me to the opportunity to give you some examples. And there are images from real RPE cells, retinal pigment epithelial cells, which do not require any other understanding, but simply to exemplify these processes I've been talking about. And the first one refers directly to the aging process. Now, we know that as we age, the production of different components essential for these various processes inside the cell decline. And we've been studying this, we as a community, for a long time, and we've been trying very hard to identify which of those components are really declining or change indeed composition with the age. And one of these, and isn't quite actually naming, is shown here in these images. And what we have here, again, we're only looking at retinal pigment epithelium that lies in the eye. So these are human sections from the back of the eye, and the intensity of redness here is in relation, or shows how much of that of a particular protein is present there. And you'll very easily notice that there is a decline as we go from left to right of these sections of this redness. And from left to right, it's increasing the age of those eyes. Obviously, we looked at much more than just the 3 eyes shown here, but I've chosen this only to give this example of how one particular protein that is made up in high abundance and therefore needed for the function of this tissue, of this monolayer of cells, decline with age. So we have less and less of it as we age. There is also peculiarity. Remember, we spoke about macular. Now, if we look at pairs of sections from the macular, the central part of the retina, and the peripheral retina, the bottom, for each one of these, so these are all pairs from the same eyes, you will notice that this particular protein is needed in high abundance more at the macular side than in the periphery. And this is true for each case we've looked at. And we've looked in science, we have to look through different methods, not just one. Yet with age, the actual decline is seen exactly at the site where this protein is needed. So we've made one example of, I've shown one example of how a protein that is very important for the function of this tissue, which in itself is essential for the function of retina, declines with age. The very same protein provides us with a different example of what happens in terms of processes that are age-related and underpin age-related decline in the eye. And that is one that will show a video here in which we'll show 2 live retinal pigment epithelial cells in a dish in the lab, where that specific protein is labeled by us with a green protein that fluoresces and allows us to see it. And the way that we look at this allowed us to see that the scope, the purpose for which retinal pigment epithelial cells make this protein is to be packed in these vesicles, which are sent into that outside space of the cell, into that filling I talked about. And hopefully this will run. And you see here a bit quicker than it happens in real life, but it'll give you a sense of how much of this protein is made of the cells and how neatly it is packed in these vesicles, which are all sent in that filling outside of the cell. So it is made in high abundance. It is made in order to be sent the outside of the cell, so its function is to protect the cells from the outside. And yet, what we also know is that in its normal form, so no defect in this protein, if we just look at one cell here, you imagine it, so it's in the background, the cell. The green, again, just shows the location of that protein, which again, it's a normal protein. You'll see here how these cells look. I'm sorry, it doesn't seem to be able to show you, but the movie would just show how a cell, it's seen from all sides with this protein of having this distribution inside the cell. I've also mentioned that apart from aging, some mutations in some proteins of importance for the functioning of various specialized cells in the eye can increase the risk of developing disease. And this particular protein is also an example of that. The change between a normal version of it. And a version that increases the risk of developing age-related macular degeneration lies only in one base of the 3 billion bases of DNA in the cell. So the difference between these two proteins lies in only 1 out of 3 billion coding bases. Let's see if this works. Yes, this would work. So this is a cell, an RP cells that makes the mutant protein, which exists in the population, and the people that have this variant are at increased risk of developing AMD, as opposed to this one, which doesn't seem to want to. Oh, now it runs. Okay, so these are the difference between a normal presenting protein in that epithelial cells, as opposed to this AMD different protein. So we hope I've given you some example and of the sort of work that we're doing. And clearly, I think what this shows is the level of detail that the research in this field has gone. We're actually looking at what individual proteins do inside the cell, and we're trying to understand how these cells function, what changes occur with aging in them, and clearly with the intention to manipulate and to see how we can intervene in doing this. Now, before I conclude, I just want to summarize what I've tried to talk about in this very, very short time. And a few ideas are here. Unsurprisingly, our eyes age. And indeed, aging affects cells with critical roles for vision. And it does so in a very dramatic way in many respects. And this is why aging is the highest risk factors for developing age-related macular degeneration. The changes that occur with aging are both inside and outside of the cells, and they affect very often the communication between cells of the same type and different types of cells inside the eye. These changes are unfortunately irreversible, and tackling the prevention, the slowing down and treatment, all of these tasks are as complex as the disease. So this is why finding a cure is taking so long time. Studying age-related changing poses particular challenges by the simple fact that we need to reproduce in laboratory age-related conditions that take a long time. So how we do that in a rational and effective way, it's obviously something that has posed a lot of problems, that we certainly have made a lot of progress. Despite all this, we have now, over the last at least decade, probably more, made really big leaps in understanding of how these very specialized and very special cells function. We made big progress in understanding how disease occurs. We saw the level. We actually look inside the cell, and it's based on these findings that we and others are trying to develop best ways of preventing, slowing down, and treating the disease. And in this respect, that Macular Society has this major role in supporting this type of research. You know, as I said at the very beginning, and I'm going back in full circle to that message, in a field that is hugely underrepresented in terms of funding for research. In the picture here, one of the Emil Carson showed here is was a postdoctoral research fellow that was initially funded by the Macular Society in my group. And it's once again that I like to use this opportunity to also express my thanks and group thanks for support that Macular Society has lent us to. I'll stop here.
Luminita, thank you very, very much indeed. That was a fascinating talk. Now, if anybody wants to pop some questions into the message function, please do so. We've got plenty of time for questions. But the sheer complexity of the organ itself, the structure of the retina, the structure of the eye, it must be one of the most complex organs in the body, I guess. And I guess that's why, one of the reasons why understanding what's happening in the eye is itself complex. It must be that looking at the structure of the retina, you describe it as fascinating. You're obviously really fascinated by it, but the complexity is astonishing, isn't it? Is there another organ in the body as complex, apart from the brain, as the eye?
Well, I honestly don't know, and I hope I mean, there will be some subjective way of thinking about this. Obviously, I'm passionate about what the function that this organ stuff allows us to. But I think a lot of people will share that, both in terms of importance, but also if you are to think really how these very different types of cells are form this organ, and indeed its size versus its importance, everything about it. I think I cannot think of anything else that would be brain. It's a very complex functioning organ, but it's much more homogeneous in terms of composition, rather than the eye.
And the other thing about the eye is that it has this enormous high metabolic rate, doesn't it? So I think it is the most highly active bit of the body as well. So there's more going on. There's more active biological processes going on in the retina and the eye than in any other organ. And so that sort of makes me, as a non-scientist, think, well, I'm not surprised it wears out quicker than other bits of the body, because it's more active, it's doing more, so it must wear itself out. It's just exhausting itself.
Exactly, yes. I mean, again, there will be, in terms of high metabolic activity, there are others in the body that are taking sort of intense metabolic activities. But in terms of sensory organs, probably the eye stands out in that respect.
And the other thing, and I don't want to take issue with the Almighty over his design plans, but it seems to me to be a bit of a fault that so much of the workings of the retina rely on a single layer of cells, this retinal pigment epithelium, just one layer of cells. We could do with a backup layer, really, couldn't we? So it makes it quite sort of vulnerable in a way, does it? Is that?
It does, yes. And of course, we now know that what's underneath it, the vasculature, is also important. And there are also changes that occur with aging there. And yes, you would wonder that nature would have supplied us, the evolution, with some sort of backup. But this is one of the quirkiness of, human development, if so. And another one being, if you think about it, in relation to the eye, this is more in relation to the vertebrate eye evolution, why this light sensitive tissue lies all the way at the back of the eye when the light enters the eye in front. Now, indeed, we know that less evolved organisms will have the light sensing cells towards the front of the eye rather than the back of the eye. And I haven't found a complete satisfying explanation for this.
That's really interesting. And I guess, I suppose, evolutionarily speaking, because we didn't, as a species, live all that long until relatively recently, the fact that we only had one layer of retinal epithelial cells maybe didn't matter, because nobody, historically lived long enough for it to be a problem. It's a new it's a 21st century problem. Maybe that's what it is. Now, look, we've got some questions, Luminita, coming in here. And one thing which is an interesting point is we know that at the moment there isn't a treatment for dry AMD, but there is for wet AMD. So one of the questions here is what does Eylea and Lucentis and these Avastin, these drugs do target proteins, don't they, or the creation, the manufacture of certain proteins in the eye? How does that work? Can you? It's interfering with a protein. These proteins are very important, are they?
Yes, and I can actually use what I said and some of the diagrams to explain this. We know that AMD comes in two forms, or usually referred as dry and wet, and it's the dry form that is most prevalent. The wet one is only present in about 10% of cases. The name wet comes from the fact that it comes with leakage of that vasculature that is immediately underneath this epithelial tissue. I have alluded to the fact that this epithelial cell layer forms a barrier and lies at the interface between the retina, the neuroretina, and that vasculature. What happens in wet AMD, that barrier is broken and there is leakage of the blood from underneath the RP into the retina. Now, blood touching any neuron type cells, it's very damaging. It's exactly the same way when any leakage of blood in the brain, if the blood-brain barrier is compromised somehow, the same sort of damage would occur in the retina, in the neuroretina. Now, what the drugs mentioned here do, they actually try to inhibit proteins that indeed lie in that environment, in that filling of the cell, in order for the new blood vessels not to be, new leaky blood vessels not to be formed. So this is why those drugs given currently in injections actually function at the back of the eye by stopping, at least for some time, that leakage of blood at the back of the eye, and therefore deterioration. They are not in any way efficient for the dry form of AMD.
No, so we're still looking, aren't we, for a treatment for the dry form of wet AMD. We've got a couple of questions coming in here over the use of dietary supplements to stave off. There is, I mean, there is this is based on the theory that as part of the aging process, oxidants or free radicals in the body and speed up the aging process, and so vitamins, certain minerals, antioxidants that we find in fruit and vegetables, for example, can slow down the aging process. Do you want to say anything about that? That's the theory of supplements, but they haven't been shown to be really an effective treatment, have they?
Yeah. It is quite natural that people have thought of tackling changes that occur or lead to AMD or to macular degeneration in general can be tackled by preventing the build-up of those byproducts, some of which are exactly the ones you mentioned, the oxidants. Biologists and chemists call them also free radicals. So these are byproducts of that very intense metabolism. So they occur in all cell types following metabolic activity. And now the problem with those, and the reason why they haven't been so successful in terms of orally taken supplements, are, if you think about it, taking something orally has to be taken in such big quantities. It actually goes, in the digestive system, then through lots of barriers in the body, and then go into the bloodstream. And with the bloodstream, a tiny portion of that reaches the actual site where this, hopefully will have some effect and have to go actually in there inside the cell. So you have to present those cells, in this case, this retinal pigment epithelium, which are very neatly tucked at the back of the eye, sandwiched between two very tight structures there. You have to present those antioxidants in the right amount and in the right form, activity, to actually be able to function as such. However, a good diet throughout life, not only in old age, is recognized as having, and I would say good diet is everything that contains antioxidants naturally, is recognized as potentially having a beneficial effect onto preventing or slowing down the progression of these age-related changes. So there's where these factors need to be balanced out.
So I think people often get conflicting advice on this. Some ophthalmologists will recommend it and in fact used to prescribe it when the NHS allowed prescriptions for supplements, and others say there's not really enough evidence to support it. So although Luminita, you're not an ophthalmologist, I understand that. Often people will say, well, there's no evidence it does me any harm, so I'll take it to be
exactly
safe.
Exactly, and I was about to say that, but obviously I'm a non-clinician, so I'm in a position to advise on that. I'm simply giving the scientific explanation of that. But certainly, yes, I mean, good supplements, good quality supplements will not do anything else. But a good diet, I would emphasize, would surely bring a lot of benefits for this and other areas.
Absolutely, yes. And what we know is that what's good for your heart and your lungs and is also good for your eyes. We know that, don't we? That's
Exactly.
absolutely right. So it's the same advice that we get from all our doctors all the time. Exercise, keep your weight and blood pressure at normal levels, eat lots of healthy green leafy vegetables, and don't smoke. So that's the same advice for macular degeneration as for lots of other conditions as well. So another question here says somebody's interested here in the nature of the RPE. The question is, how archaic is the RPE, and is there anything to be learned from the non-vertebrate eye? Now, we always say that animals don't provide us with a very good model for macular disease anyway, because most of them don't have maculae at all, do they? So what could we learn from a non-vertebrate eye, do you think?
Right, hi. I can. It is a fascinating area in terms of looking at how the eye develops and offers, if not a functional explanation, at least an anatomical explanation, how, from something that is called optic cup, when the embryo is really in early stages, how it develops and forms later the eye. It's probably fair to say that the less evolved an organism is, the vision is not that important of a sense, and therefore vision is not really developed. So this very intricate structure that humans and indeed vertebrates have of their eye, it's something that has evolved together with our evolution. So it's probably one of the best examples of that.
Another question here about, looking at the complexity of the eye and the challenges that they're presented by the disease. A question about transplant, whether there's any opportunity for transplanting healthy retinal tissue into diseased retinal tissue. We know that there's some work on stem cells, but what about transplanting bigger sections of the retina?
That would be very, very challenging. Like with any transplant, well, there are a few issues here to talk about, and just very briefly. Transplanting tissue from a different individual obviously poses the usual problems for transplant, rejection, and so on. There are different approaches to transplantation, both to look at developing good, well-functioning light-sensitive cells, photoreceptors, and developing this retinal pigment epithelium in a manner that is well functioning. There are some promising results in that area. There are not yet in any way approved treatments for such. I seem to remember that was a very good talk given in this exact series, or maybe the Macular Society, which again can be found on the Macular Society website, by Jim Bainbridge. He explained in sort of another 25 minutes or so exactly what the approach is in terms of transplantation is. So I would highly recommend that for a more comprehensive type of explanation, recent one, where we are at present with those type of approaches.
It is very challenging because we've seen the complexity of the structure of the retina, and to get all those different types of cells connecting up and working as a whole organ is a very challenging thing, isn't it? And another question here about if I know this is a clinician question really, but people who have disease in one eye and the other eye is okay, why would that happen? And is the second eye going to be affected? And I think we know that there is it's not inevitable, but it is usual, I think, for the second eye to become affected within perhaps 4 or 5 years. Is that roughly right?
It is, and again, it's only roughly because we don't have the answer to that, and I wish what we as a community would know that. Yeah, it's not known, and I wish I could have an explanation for that. I was not able to offer anything sort of that is rational from a scientific point of view why that happens.
So finally, just before we wrap up, then a question that came in earlier about, so what's the chance of us being able to halt deterioration in this? We can't turn back the clock. We can't stop ourselves aging. Is there a chance, do you think, of being able to find a way of stopping the deterioration?
I will definitely say yes, as a scientist that is enthusiastic about their work and knows the major progress we have made in terms of understanding of what's going on and understanding these processes. At the same time, I would urge caution, simply by even looking at the aging process. I try to emphasize here, it is unfortunate that we cannot say something will happen in a very short time frame. But certainly, I think I like to believe that in our lifetime, we will be able to have at least major breakthroughs that will be on that way, and hopefully, yes, beat macular disease.
Luminita, thank you very, very much indeed. That was a great way to end, great out, as they say. A question has just come in about gene therapy. We are out of time, but I promise that in the new year we will have a session on gene therapy, entirely on gene therapy, and we'll come to your questions then. Luminita Paraoan, thank you very much for being our guest tonight on the virtual clinic. It's a great pleasure to have you and listen to you. This is the final virtual clinic of 2021. We are going to be resting in December. It's very close to Christmas, the next scheduled tape. So our next virtual clinic will be on the 18th of January, 2022. Can you believe? 2022, the 18th of January. And I do hope you'll be able to join us then. Luminita, thank you very much once again for being with us tonight. And may I be the first to wish you all a very happy Christmas. See you in the new year. Thank you very much indeed. Good night, everybody.
Thank you all. Good night. Good night. Thank you.